Hi everyone, my name is Yizhu. I'm a global product manager in Miltenyi Biotec, taking care of the sample preparation portfolio. Today, I would like to talk about how to start smart with your sample preparation by focusing on the isolation of immune cells in mouse tissue. So first, there are two parts of today's talk, and in the first part, I will talk about how to get the viable single cells from mouse tissue, and by focusing on different aspects. And also the second part, we will talk about more into detail, like how to analyze the cells tissue-derived or resident immune cells in different tissue, such as propria or lung or brain or even liver. So let's start with the first part. So when you are working with tissue, mouse tissue especially, normally you might get like mouse spleen or lung or intestine, liver or brain or even tumor. But today we will not focus on the tumor. If you are interested in tumor to isolating the cells in the tumor, we have another webinar in MACS Academy you can join later. So if you are working with mouse tissue, so normally you would like to get the cells from the tissue by tissue dissociation. And in this case, we call the whole procedure sample preparation. And this sample preparation, the goal is to get single cells out from those solid tissue. And later on, after you get the single cell suspension, you might go for the downstream applications such as culturing the cells or analyzing the cells by flow cytometer, or you can even extract DNA or RNA from those cells to do molecular analysis. And you can even sort those cells out. And moreover, you could also combine with microbeads technology to isolate the cells by using, for example, CD45 microbeads or CD4 microbeads to enrich the immune cells and further to do the analysis. So today, our aim is to talk about the sample preparation. So to have a good sample preparation for immunological study, you need to focus on 4 different aspects. First, we will talk about how to proceed the consistent and efficient tissue dissociation. By doing so, then you can get higher cell viability and yield. And after you get the cells, and we will talk about how to have a good quality of the cells for your downstream applications like flow analysis. And then we will also talk about how to remove the cells debris from certain tissue samples. So first, let's start with the consistent and efficient dissociation. How to achieve to have the consistent and efficient dissociation? So if you are working with mouse, you must have experience of working with mouse spleen, for example. So when you are working with mouse spleen, normally people just mesh the spleen to get the T cells or B cells out. And this mesh we call mechanical disruption. The goal is to break down the tissue into smaller pieces and release all the cells out. But you might also experience that different people, when they are meshing the spleen, might get different results. And this creates a variation during your downstream applications. Our goal is to keep this mechanical disruption consistently, and you don't introduce variation in your experiment. But sometimes the dissociation, the pure mechanical disruption is not enough. Sometimes you need to combine with the enzymatic digestion, because the mechanical disruption is just break down the tissue into smaller pieces, and sometimes the cells are very deep into the tissue. You need to have the enzyme to digest those tissue further to break down them into single cell suspension. And this entire process we call tissue dissociation. In Miltenyi, we have developed a fully automatic instrument called gentleMACS Dissociator, and this instrument enables us to have consistent tissue dissociation without introduce any human errors or human variation. By working with this instrument, you need to combine with the gentleMACS tube, which apply the mechanical disruption, and also our tissue dissociation kit, which provide the enzymatic digestion. So let's go more into detail how this gentleMACS tube works. The gentleMACS tube is to provide the tissue dissociation, the mechanical force to dissociate the tissue into smaller pieces. So how does that work? It has a rotor and stator area. So when the gentleMACS tube is working on the machine, it will rotate in the clockwise or anti-clockwise direction, and these will break down the tissue into smaller pieces. But a very special part of this tube is actually the spacer in the tube. Because of this spacer, only the tissue can be disrupted by this rotation, but the single cells which was dissociated in the tube, they can pass through safely in the tube. This enables us to get high amount of the cells and also to retain the cell viabilities. In combination with the gentleMACS tube, we also have the tissue dissociation kit. And this tissue dissociation kit, they are specific for different type of tissue. So for example, like a spleen dissociation or lung dissociation. And each different kit, they have a defined program written on the gentleMACS dissociator. And combining all these three parts, you can do the tissue dissociation in one go without further developing your protocol. So how does this exactly work? In the gentleMACS tube, when you are doing the dissociation, what you can do is just take the tissue you would like to dissociate, put them into the gentleMACS tube and add enzymes and place the gentleMACS tube on the machine and also at the heater unit. This heater unit can heat up to 37 degrees and just press the button on the gentleMACS, then the dissociation could be done automatically. So the gentleMACS Acto with heater has 8 different positions. These 8 different positions can run different programs at a different time, or you can also run 8 samples at the same time with the same program. So what you need to do is very easy. Just put the tissue into the tube and adding the enzyme and place on the machine, press run, and after a few minutes you can get your dissociated tissue with single cell suspension. After we talk about how to get the consistent and efficient dissociation, now we would like to talk about how to improve the cell yield and viability. So many of you might already work with mouse spleen or lung, and many people might just simply apply the mechanical force to dissociate the tissue. As I said, sometimes it's not enough. Sometimes you need to combine with the enzyme to increase the cell yield. Let's look at the example of mouse lung dissociation. If you dissociate the mouse lung by combining with the enzyme, you can see you would be able to get a high amount of cells by just looking at forward scatter and sideward scatter on the flow dot plot. But if you only use the mechanical disruption, it's very difficult to get high amount of cells because a lot of cells are actually deep into the tissue, and by simply cutting the lung is not enough to get a high cell yield. And also, if you look at leukocyte CD45 positive cells, you can see there are nearly no cells when you only apply the mechanical force. This phenomenon is even more prominent when you're interested in the tissue type of cells like endothelial cells, which is CD31 positive, or epithelium cells, CD326 cells. And you can see if you only use the mechanical disruption, you nearly can't get any epithelium cells. And the yield is also related to the viability. So because of the leukocytes are more robust, when you are applying the mechanical disruption, you normally need to apply much higher mechanical disruption to be able to reach a similar amount of the cells when you compare to enzyme digestion together. But the viability of the leukocyte might not change too much. But for some other sensitive cell type like endothelial cells or epithelium cells, the viability might change dramatically. So if we look at the absolute cell count, you can see when you only apply for the mechanical force and you can get really low amount of cells. By just adding the enzyme, then you can increase the cell yield by more than 10 times. Therefore, it's very important to have both mechanical disruption and enzyme digestion all together, and gentleMACS dissociator is exactly designed for these two purposes to get the good tissue dissociation. So after you get the cells from tissue. Now we would like to talk about the quality of the cells. As you know, in the immunological study, most of the people might use flow cytometer to detect the cell type, to see what kind of cell type we're getting from the solid tissue. By using the flow analysis, you need to have an antibody to bind the surface marker of the cells. And those surface markers are epitopes. So the antibody can bind the epitopes. And the epitopes are proteins. But those proteins might be digested by the enzyme you use for the tissue dissociation. So how do we find the balance between digesting the tissue and digesting the epitopes? So in our dissociation kit, we actually select the very pure enzyme, because if the enzyme are pure, the chance of cutting the epitope unspecifically is much lower. Therefore, in our kit, we only select the pure enzyme, and to those enzymes which has a really low tryptical activity, which cannot cut the surface epitope unspecifically. For example, if you are dissociating the tissue, and here you can see a very nice population of CD8 and CD25. However, if you use less pure enzyme, so like crude enzyme, and they might have a contamination of trypsin or other aggressive enzyme, as you might know, the trypsin will cut everything. So then you don't see CD8 population, you also don't see CD25 population, and you might create false negative result in your analysis. When you see the data like this, it doesn't mean those CD8 cells are not in your tissue. It's just because the epitope are digested by the enzyme unspecifically, so you cannot really detect those cells. Therefore, to know which epitope could be cleaved would be very important before you do experiment. However, it's not possible to find the enzyme which don't cut epitopes at all. So in our dissociation kit, we actually test more than 200 of epitopes to see which epitope are sensitive to which combination of the enzyme. In our QC process, we always detect which epitope is sensitive, and we know in advance which epitope we have to be careful if we use which enzyme combination. This would be also very important information for you when you are doing tissue dissociation, because you need to know which epitope can be detected afterwards. The last part I would like to talk about is to remove the cell debris. So some of the tissue are very important to remove the cell debris, but some of the tissue are fine. So it really depends on which tissue you are working with. So we have a ready-to-use debris removal solution which you can remove your cell debris in 20 minutes. So how does that work? So you just mix the cell with debris removal solution and overlay with PBS and do a very quick centrifugation with 10 minutes with full break. And then after centrifugation, you can get the cells and debris in the lower two layers. And you just remove the debris and resuspend and wash the cells. Then you can get the debris-free cells suspension. And what type of tissue would be important to use the debris removal solution? For example, like liver or brain or spinal cord. Because in the liver, when you are doing liver dissociation, most of the cells are hepatocytes. They die immediately after you do dissociation. So it would be very important to remove them. And also in brain, there are a lot of myelins, and you need to remove them before you do the dissociation. If you don't remove the debris, those debris might bind to the antibody unspecifically, and you might create some false positive results. So let's see, for example, if you take the brain without removing the debris, you can see the debris are very prominent here. And after removing the debris, you can see those debris are gone, and then now you can again see your cell population. Without removing the debris, you nearly don't see where the cell population are. So it is also very important to remove the debris before you do any downstream applications. So we already talked about the important aspect to dissociate the tissue into single-cell suspension. And next, I would like to focus more into detail how to analyze the tissue-derived or resident immune cells in different tissue, such as intestine, lung, brain, or liver. So in the first part, we already talked about how to generate single-cell suspension from solid tissue, and these are the input cells you will get. And normally people might go for the flow analysis to see what type of the cells you get from those tissue. And you can also combine with magnetic cell separation to pre-enrich the cells before you do flow analysis, and those will be your readout of the experiment. Normally, we would use the surface marker to detect those cells, such as CD45 for leukocytes, or CD3, CD4 for T cells, and CD11C for DCs, or FOAT for macrophages. In this part, we will talk about 4 different tissues like intestine, lung, or spinal cord, brain, and liver. So first, we would like to talk about intestine. Before going further, I would like to introduce you about the structure of the intestine. This is, for example, like small intestine. They have villi in the intestine. They consist of many epithelium cells, and especially the small intestine, there's a very special structure called Peyer's patches. So they are like a lymph node in the small intestine, and they consist of newly generated immune cells. Normally, when you dissociate the intestine, you will remove the Peyer's patch in advance by simply just cutting them out with scissors. And there are also a lot of lymphocytes. They are just sitting in between the epithelium layers. Those are called IL, called intraepithelium lymphocytes. To obtain those cells are pretty easy because you can just treat the intestine with EDTA to release the conjunction between the epithelium cells, and those cells, those IELs, will be released into the solution, and you can get them very easily. The last compartment we will talk about is the lamina propria. There are also many lymphocytes sitting in this compartment, but if you want to get the cells from this compartment, you will need to treat with enzyme to digest the tissue completely and release the lymphocyte from this compartment. And why are we talking about these 3 different compartments? Because the composition of the immune cells in these 3 compartments, they are completely different. So when you are doing tissue dissociation, it's better to separate these 3 compartments and to analyze them separately. So when you are working with intestine, small intestine or colon, the important thing is to dissociate the tissue with Lamina propria dissociation kit on gentleMACS, and then further down you can analyze them with flow. The tip here is you need to wash the small intestine by predigestion solution. And those predigestion solution steps are very important to prepare intestine because intestine contains a lot of mucus and you need to use DTT to remove the mucus. Otherwise, the mucus might affect the efficiency of the enzyme digestion later on. And also you need to treat the intestine with EDTA to release all the epithelium layer and also IELs to make sure that you don't have the contamination of the IELs in the lamina propria layer. And very important is to work fast, because the predigestion step may take you more than an hour to complete and the cells might die in between, so it's very important to work fast to increase the cell viability. Normally people might work with colitis models because in the colitis model there are a lot of lymphocytes infiltrating into colon and you will be able to get a lot of immune cells from mouse colon. So let's have a look. When you are isolating the cells from intestine, you can see, for example, in the IEL compartment, there are much more CD8 T cells, but there are not so many in the lamina propria compartment. But in the lamina propria compartment, there are more B cells instead of in the IEL fraction. So therefore, it's very important to separate these 2 different fractions. So then the next, we will talk about how to dissociate the mouse lung. For mouse lung, it's also very easy. So you just take the lung lobe from the mouse lung and without cutting them further, just put into the gentleMACS tube together with lung dissociation kit. So press the run button, then afterward you get single cell suspension. Then you can do flow analysis afterward directly. In here, we would like to introduce more about how to get the dendritic cells specifically, because it works also very nicely together with CD11C microbeads. Together, you can enrich the dendritic cells for further analysis. The tip here is because there are many blood vessels in the lung, if you would like to distinguish between the circulating leukocyte and the tissue-resident leukocyte, it's better to perfuse the lung before you proceed with the tissue dissociation. This perfusion we are talking about here, it's a very easy perfusion. You just perfuse out all the leukocyte which retain in the blood vessels. And as I said, if you work with gentleMACS, you can simply take the lung lobe directly without cutting the tissue further. So if you dissociate the tissue, the lung tissue, you can see you can get around 5 to 7, 10 to the 6 CD45 positive cells in one mouse lung, which is quite a large amount of cells. Because of this high cell yield, you will be able to pre-enrich the CD11C positive dendritic cells further down. So you can see after dissociation, there's around 20% of the CD11C positive dendritic cells, and by enrich with microbeads, you can enrich those cells up to 95%. And further down, you might do flow sorting or cell analysis for the subpopulations. There is also an application note available to describe more detail how to do the experiment. If you are interested, we can send you after the webinar. So further, we would like to talk about how to dissociate the brain and spinal cord. So for the brain and spinal cord, it's very special because there are many myelins in the brain and spinal cord, and these might affect the cell analysis later on. So it's better to combine the debris removal step to remove the myelins before you do any downstream application. And also here, we use the multi-tissue dissociation kit 1 because the most common enzyme to use for neuron tissues, they might cleave out many epitopes on the immune cells. If you are working with brain or spinal cord, it would be recommended to work with inflamed neural tissue because in the normal mouse brain, you actually don't get so many immune cells because of the blood-brain barrier. So it's better to work with the, for example, like EAE models. And also because there are many blood vessels in the brain, so if you would like to distinguish between the circulating leukocyte and the resident leukocyte, it's better to perfuse the brain before dissociation. And as I said, the epitope sensitivity and removal of myelin would be also very important in this case. So let's have a look. When you are dissociating the brain or spinal cord, the common enzyme which used to dissociate this tissue normally contains papain or trypsin. And those enzymes could potentially cut out the CD8. Then you won't be able to detect the CD8 in the brain or spinal cord. And also they might cut out the CD19 or B220, which you won't be able to detect the B cells in the brain or spinal cord. Therefore, in this case, we will recommend to use the multi-tissue dissociation kit 1, which completely preserve those epitopes. And in the protocol, you can find also many antibodies we recommend to work together with the multi-tissue dissociation kit 1 and to analyze your immune cells further. So the last part, we will talk about the liver dissociation. The immune cells in the liver, which is very popular, is called Kupffer cells. And those Kupffer cells, they are F480 positive cells. And in this case, we will recommend to use also combination of the debris removal to remove the dead hepatocytes and also to isolate directly the Kupffer cells by F480 microbeads. And in the liver dissociation, normally people, when they are dissociating the liver, they use perfusion method. But in this case, because we are only focused on Kupffer cells without focusing on the hepatocyte, so you don't have to use perfusion. You just have to take the liver out and put them together with the liver dissociation kit on the gentleMACS. Then you can get all the non-hepatocyte cells. And because the hepatocytes are very fragile, they will die with any mechanical disruption. And so therefore, they become debris and you have to remove them before you do any downstream applications. So after removing the debris, you can see the Kupffer cells is around 20% among the CD45 cells. And you can use a direct microbeads called F480 microbeads to further pre-enrich those Kupffer cells up to 98%. And you could potentially analyze the subpopulation of the Kupffer cells or further to sort them out. So here, after talking about the tissue dissociation and also giving you some example of how to work with different tissues, the take-home message here is if you want to have a good sample preparation, it's very important to have a consistent and efficient dissociation, especially with gentleMACS technology. Because the method is very gentle to the tissue, you will be able to obtain high cell viability and yield. And most importantly is to know your epitope before you do any downstream application to preserve the epitope to have a good quality of data. If you are interested how other people are using the gentleMACS technology, you can go to our website to download this reference list. In this reference list, you can find different people are working with different tissues, how they are doing their downstream applications, and how they are analyzing their cells. And also, if you are interested in how to do the dissociation with gentleMACS dissociator, you can go to our Miltenyi TV. In the tutorial part, you can find the video protocol. And those video protocols show you step by step in a very quick path how to dissociate Lamina propria and mouse lung and mouse tumor. And in the next month, we will also release a new protocol for mouse spleen. So thank you very much for your attention.